Pedro Mendes is a professor of cell biology under the supervision of
So I apologize already for those of you who were on the workshop on Monday, because some of this will be repeat, but hopefully it will not be too. And then the second part, I want to talk about an application of the software, and that's actually some of the work that I'm carrying out in my group. So I will divide the work in a few parts. So the first one will be essentially introducing the software. Then I will talk about the application area, which is iron metabolism, iron physiology. So I'll give a little bit background. Some of you might not be so familiar with it. And then I'll talk about constructing a model for mice, and then going from the mouse model towards a human model, and then later on to try and see if we can make, rather than having a generalized human model, if we can have models for specific individuals. So personalizing models. So as Rodrigo already pointed out, I started my career as a young researcher, and I wanted to do simulation of biochemical networks, and there was no software I could use on my computer. There was one that existed, but it was for a different type of computers. In those days we had personal computers, and there were different kinds. Okay, so someone made software for an Amiga, and I had a PC, so I couldn't use the software from the Amiga, so I thought, okay, I can do this. It's not so difficult. Okay, so many decades later, I'm still here telling you it's not very difficult, but it's really an ongoing work. It was a little bit more difficult than I thought first, but you know, if you don't make things, they don't happen. So that's what I did. I wanted to do the simulation, so I ended up spending half of my career doing the software. And that software was called Gepazi. It is actually still in existence. People can download and use it, and some do. I do not encourage that, because actually the software that we now make called Gepazi does everything that Gepazi does, and hopefully quite a little bit more. But the other one is still available. It's open source, such as Gepazi is. So what is Gepazi for? By the way, Gepazi is a name. People like to ask whether does the name come from. And names are just names. They don't really mean a lot, but we had to make one, and we tried to make one that didn't exist in any other product or language, etc. Spend a lot of time thinking about what should it be called, just so if you Google it, you find our product, not somebody else's. And it turned out that at that time, Gepazi was really a unique word. There was not really any Google hit, so that's what it is. It stands for Complex Pathway Simulator or Collaborative Pathway Simulator or anything you want. It's really called COPASI. So what the software does is that it allows you to represent a biochemical system. It can be a set of reactions that happen inside the cell, or it can be reactions that happen over all the body, as you will see later on, and represents that model with different mathematical formalisms. So the software really focuses on the biochemistry. It is more directed towards the biologist and to allow them to represent what they want in the way they're used to. So there's really no end, people don't enter differential equations, they don't enter probability functions, but those things are what underlie the model, right? So the software itself builds the model. It asks the minimal amount it needs, then build the equations. Those equations can be of three different formalisms. The first one is the most commonly used. It's based on differential equations. And in our case, we don't represent space. We don't represent diffusion. Therefore, it's a set of ordinary differential equations. And the software builds them itself. Now, you can also, and it's very important many times when you have systems that have very small number of molecules, such as, for example, when you have transcription factors and genes. There's usually 2 copies of genes in our genome. Sometimes they get copied more, but for most proteins, there's only 2 coding copies. So that's number 2, okay? Therefore, you can have 1 copy active, 2 copies active, but you cannot have 1.5 copies active because there's no such thing as half a gene. So in those circumstances, it's important to represent the system as a probabilistic formalism because noise is very important when you have very small numbers. And also, it is a system that is no longer continuous. It's continuous in time, but it's discrete in the number of particles because you can only have, as I said, integers. And that is the formalism that people normally call the SSA, the stochastic simulation algorithm, also known very often by the name of the person who developed it, called Gillespie, Dan Gillespie. So those are the 2 main formalisms, and just very recently we added a 3rd formalism, which is stochastic differential equations. That is, we allow you to add noise terms to the differential equations. Now there is, because of the underlying chemistry of the system, it is not unrestricted noise that you can add because it has to be done in a way that does not violate mass conservation, okay? Even though things are probabilistic, they still obey the laws of physics as we know it, so noise can make things faster or slower, but it cannot create molecules. One molecule reacts because one more reacts because of noise, then we need to account for it in both sides of the reaction. There is a special way of adding this, and the software does that for you. So the user, as a biochemist or a biologist, all you need to do is to say, this reaction has this noise term. The system takes care about not violating mass conservation. Then it has a number of other methods to simulate based on these 3 formalisms. So we have differential equation solvers, we have the stochastic simulation algorithms, we have an SDE solver. Also, by the way, all these systems, for those of you more on the mathematical side or numerical side, most of the times these systems are very stiff, so we cannot just use simple algorithms. We have to use algorithms that are stiffly stable. And that for the ODEs is not so much a problem. Things have been dealt with for a long time. But for stochastic differential equations, it's not as simple. There are not so many algorithms we could have picked. And so there are then a number of analysis methods that we also provide, such as sensitivity analysis and optimization and so on. And I'll talk a little bit about each of these go along. Now, I need to acknowledge my colleagues. COPASI, as I said, one of the interpretations for the CEO is collaborative. So it's no longer a piece of my work. I'm one of the contributors. COPASI was just myself, but now it's a lot of colleagues. And in particular, there's 3 major groups working on this. One at Virginia Tech, led by Stefan Hoops. Another one in Heidelberg, led by Ursula Kummer. Now, Ursula Kummer's group members, some of them have become now junior group leaders, so I have to acknowledge their groups as well. So Jürgen Pahle and Sven Sahle have their own smaller groups. We have altogether, at the moment, 3 people working full-time on the software. So we have 3 full-time software developers, and then the number of the rest of us working on it a little bit more part-time, because we have to write grant proposals and review articles and interview students, grade them, and so we cannot spend all our time on the software, but we do spend as much as we can. And we have funding, we have obtained funding for this software fortunately from a number of funding agencies on all of the 3 or 4 countries where we have been. So previously I was also in the UK, so I have received funding from the UK research councils, also now from the National Institutes of Health, and Ursula Kummer has obtained funding from the German Ministry of Science and also from some private foundations. We're very grateful to them, because without this funding we couldn't make the software for anyone who uses it. So how does Copasi fit into the whole scientific process? This is a diagram that kind of summarizes what we do as scientists. Even those that don't use computers, they might not have the simulation part, but they will have all the rest. Okay, so they do experiments, they collect data, they have to publish that data, they have to store it in databases, they have to build models either explicitly or implicitly, either as equations or just a diagram in figure number 7 for their papers. And they have to obtain that by collecting information from the literature or also from this body of knowledge we call, fuzzily called knowledge, which I put there as a little cloud because we don't have it very well defined. Now, Copasi sits in, let's see if this... I'll point to it, it's okay. So in Copasi works mainly on that cycle that you see on the top right, where you go from a model and a set of parameters, and with that, what we normally call simulation, which there is written as forward modeling. Given a model and its parameters, we can determine the system. We can determine a simulation of the system, so an evolution of the variables of that system, so the concentrations of the various chemical species. And that's what we call simulation. Now, Copasi also works on the reverse, on the... So we go from model and parameters to the simulation. So this simulates an experiment. But on the other hand, we may also have collected experimental data from experiments, and now we want to compare it to our model. And we want our model to be as representative as possible of the real system that we have probed with some experiments. And for that, we do something called inverse modeling. So we compare the two and we improve the model. And that some people will know that as fitting, or parameter estimation, or inference, or regression. There's many names for it, but it's all the same. In this case, it's nonlinear regression of ordinary differential equations. And so Copasi sits right in the middle. It allows you to do both forward and the reverse. So as you may have realized, or any modeler have thought properly about it will have realized, modeling, or in fact all of science, is based on this question: how much do the parameters of the model affect the variables of the system? And when you do experiments, you ask exactly the same questions. If you want to know what does a transcription factor do, you're really asking about how much, what is the strength of this transcription factor in inducing the expression of a certain gene. So there's a parameter there that you want to learn, and then there's a variable that you want to see how it affects. And everything in modeling is basically answering this question in one form or another. And I just give 3 examples there of questions that really don't seem to be the same thing, but are all eventually the same thing. So for example, how do you design a drug to cure a disease? Well, you want to find a drug that affects a parameter which has a large effect on the disease and therefore brings back to normal. It can also be what properties of my model, what properties give my model robustness. It's the same thing. What is a robust model is one that doesn't really sense much, is one that is not very much affected by changes in the parameter values. So it's answering the same question. Or finally, for example, which parameters do I need accurate estimates? So doing an experiment, you cannot determine all the parameters. You don't have funding for everything. It's not all practical. There's no time, there's no money to do everything. So the question becomes, which ones should I focus on? Which parameters should I focus on? And those are also the parameters that have high sensitivity, by the way. So this is really all we do. And all the methods in Copasi try to answer this question in one way or another. This slide is just to show the formalism behind the ODE approach of building these models. I'm not going to spend much time on this, but essentially just to say that this is sufficiently formal that the software can build the equations itself. These are nonlinear equations, but they're actually linear combinations of nonlinear terms. So they have quite a bit more of structure. As you can see here, the differential equations are essentially an algebraic sum of a certain set of terms, and those terms happen to be nonlinear. Okay? So there's actually properties that come from the linear combination, which our software also works on. For example, mass conservation is one of them. But a lot of it, of course, gets complicated when you have these nonlinear expressions. For the biochemists in the audience, you will probably recognize that this here is what we normally call an enzyme kinetic rate law. And so those are the only things that you have to supply the software. And in fact, we have a number of them already pre-coded. So you can pick from the most popular ones, or if not, then you might have to enter some equations. But those are what the biochemists already are doing when they're analyzing their data. So we're not asking them to write differential equations. The differential equations are all generated automatically. Now the software then can do what we call time course simulations. These can be deterministic when they use the ODE formalism, such as there. This is, by the way, exactly the same model. Or they can be stochastic if we use the stochastic simulation algorithm. And now also using stochastic differential equations, they would also be stochastic, such as that. And that's kind of the most basic use of the software, and this method is underlying a number of others. We can also do complex simulations where essentially there are a sequence of different simulations, and we call that parameter scanning. If we change parameter values regularly along a grid, or if we just get values for them from some distribution, we call that parameter sampling. And again, for a certain model, there's two examples. This is what you do. The top one is parameter scanning, where you're changing, in fact, in that case, two different parameters regularly in a regular set of values. And here is the same two parameters, but changed, drawn from a random distribution. And in fact, as you can see, the two curves reflect the same thing, but they reflect it in different ways. So it depends how you want to approach studying your system. So it allows to do that. It also does calculations on sensitivity analysis. So I think even people from the electrical engineering side will know what sensitivity analysis is. It's essentially answering the question I just mentioned. How much does a parameter affect a variable? Okay, in sensitivity analysis, that is done using a partial derivative. So essentially, you ignore that the curve is nonlinear, you get the tangent at the point where you want, and that's the slope of that tangent. It's a partial derivative. We can calculate that, and also including a formalism known as metabolic control analysis that recognizes some special properties of these systems, and from there, it generated a number of theorems that allows us to calculate things. Now, one thing that I particularly like myself is optimization. And again, this might be something that people from the machine learning side might recognize as well. Essentially, what regression is what you in machine learning would call training your model. It's essentially regression. It's fitting the parameters of the model such that it represents our input data, such that it can produce an output that matches the same output that in experiments you have for the equivalent input. But because these systems are nonlinear and very often badly behaved because they're very stiff and they have many times multiple minima, we have to use various different types of optimization algorithms, and we have included quite a very large number of them. As you can see, there's a list there. This is a menu, and those algorithms, if you can read them, not too small for you, you'll recognize some of these things as being Levenberg-Marquardt algorithm is the classical algorithm used for nonlinear regression in estimation, in statistical estimation, but then you will also recognize things such as genetic algorithms or particle swarm optimization, simulated annealing. And these are algorithms, actually many of them inspired by nature, so fit very nicely this conference theme. But they are algorithms that will attempt to find global minima, or at least they have a chance to find global minima. So we have a menu of these, and for those of you more from the computer science side, why do we have a menu is, of course, because of Wolpert and McCready big insight in the late 90s of the fact that in global optimization, there is no best algorithm. It's a problem-dependent case. And so for a specific set of problems, there will be a better algorithm, but for a different set of problems, there will be another better algorithm. In fact, there seems to be a set of problems, which we don't know what they are, for which random search is the best algorithm. That's actually what they proved. So from that point of view, giving our users a chance of having different algorithms is the most robust thing we could do. And we have a number of them, and we keep including new ones. So every time a new algorithm comes up that looks promising, we add it because, particularly if it's of a different kind, of a different approach than we have. So there's a list of them, as you can recognize some of algorithms there. So in optimization, we allow the users, just to go a little bit backwards. In optimization, we allow users to define any kind of arbitrary objective function, cost function or energy function, whatever you want to call it. That could be as simple as maximizing a concentration of a certain chemical, or as complicated as minimizing a sum of eigenvalues of the Jacobian matrix. So you can do quite a lot of things with this optimization. One of the things you can do, which most people end up doing, is parameter estimation. So in that case, the optimization function is really a sum of squares. It's a least squares function. And now we're changing the model parameters in order to try and fit the data as best as possible. And we have all those same algorithms available for people to do parameter estimation. Now, Copasi is essentially a generic tool, and we make it, well, I started making it because I wanted to use it, as I told you. But of course, other people do want to use it, and eventually our tool has gotten quite a bit of adoption around the world. We have a page on our website, this one here, which actually we list all the papers that use Copasi. All the papers that use Copasi and cite us in such a way that we can find out that they did use Copasi, which I think is most of them. And Google Scholar is pretty good. It even finds master's theses and PhD theses, so we have all of those listed. It pays off to cite us, because then we will put you on the map. And I would just point out that up to 2016, there was nothing in Central America and also not in Costa Rica. Now, we haven't updated this. We have some 2017 data. We have not fully done 2017, and we don't have any data from 2018. So maybe there are already some papers out there from here. But if not, I would really like to put a dot on the map in Central America there. And hopefully Costa Rica will be the first. We have a number of publications. I'm not going to talk much about it. Some of them are generic, just describing the software. Others are about particular algorithms that we have used. For example, we use one approach to do stochastic, not really stochastic simulation, but to estimate variance of stochastic systems when you have large numbers of particles, called the linear noise approximation. So we have some papers describing how we implemented linear noise approximation, etc. So those are available, and people. So this will end my part of talking about Copasi. Time to actually ask, what am I doing with time? Am I halfway? Okay, so more or less, okay, in time. So just to finish off Copasi, what are we still working on? Because of course we have to justify why we get funding. Most of the time, I have to say, our programmers spend time supporting users. And that's developer, just so we can help all the people that are using the software and fix all the bugs that appear, etc. So we always are adding new features as well, and those are the ones that we use to justify the funding, essentially. So what is coming in the pipeline? Okay, so right now, one of the major methods we're working on, that we hopefully will release sometime soon is delay differential equations. Currently, we don't have delay differential equations. Turns out that it's very hard also to do delay differential equations when you have stiffness. When your system is all more or less on the same time scale, then that's no problem at all. There's many methods to do it. But with stiffness, there's only one that we know of. We're implementing it. And there are some issues with it that we will come up at some stage. We're also adding model management tools. Those are tools to actually help people manage the model. The model now seen as a data object. And the model itself, if you do modeling like I do, you end up with folders full of models with very long names because you encode all the information. All the metadata is encoded in the name, and that's very suboptimal. From those of you from bioinformatics will know, big problem with data is actually managing it. It's the biggest problem with data. And so we're looking at models as data objects and starting to add a number of management issues on them. And the two major ones are versioning models, so allowing users to create several versions of the model so that they can go back and forth, a little bit like you do in code versioning systems, except that code versioning systems don't work for this. People have tried, and they really don't. The second thing is also to allow users to register the provenance of the information that builds the model. So if you're going to add a parameter value that you obtain from the literature, you should be able to formally annotate the model to say, it came from this paper, and maybe you update it later on and you say, oh, well, now I have a better estimate from that other paper, or it came from a database, or whatever the source is. So we would like to be able to help people track of how they build the models and how the models evolve. That's actually one of the hardest things. It's a lot harder than to implement differential equations. Front end is the hardest thing. The second hardest thing is managing the data. The easiest is actually the numerical methods. And we also want to start improving how very large models are handled. They're talking about models that have, say, 5,000 reactions, 10,000 reactions. These come from a field called whole genome metabolic networks, or now some people are also doing models for whole cell models that include everything. The transcription of all the genes, the translation of all the proteins, protein-protein interactions, you name it, the whole stuff in there in a single model. So those models have very large number of reactions. And it turns out that some things in the front end slow down when you have tables with hundreds of thousands of parameters and things like that. So we have to kind of implement methods to help managing the front end. Essentially, the front end is the biggest problem. Okay, so now to switch topics completely. I will actually no longer explicitly talk about software anymore. And in fact, maybe a little bit about equations, but very, very little. So I'm going to talk about how I use Copasi in my group to address a problem in biomedicine. In fact, a problem I would say in human health, not in so much in disease necessarily, because we really want to understand normal body rather than just the diseased body. This has to do with iron physiology. And you may or may not be aware of iron, but iron is really very important. It's absolutely necessary for all life. All living organisms require iron. It is a ligand of the most primitive enzymes. If you do phylogeny of enzymes, you find the oldest enzymes. They are all, or most of them have iron-binding proteins. So iron has been around for a long time. And the reason for iron being so necessary, apart from the fact that it's also one of the most abundant elements in the universe, but we don't care about stars, so we really want to see how it works in the body. It is the fact that it's a very flexible transition metal that is able to exist in different oxidation states in stable form, in solution. There's not many other, I mean, there's a few other that are like this. Copper is another one, but somehow copper is not as widely used. Actually, a little bit more dangerous than iron. I'm going to tell you about the dangers of iron, and probably the reason why copper is not used is because it's even more dangerous than iron. So most likely life avoided using copper as long as it can, only for the things that it needs to use. Why is iron important? It's for two major reasons. One is that it's the best ligand for oxygen. So anytime oxygen is transported across the body, there is iron involved in that process, or reactions where there is oxygenation molecules. Again, iron is going to be included. For example, cytochromes P450 are all iron ligand enzymes. And it's also very useful for electron transfer. So you find iron in many places in the electron transport chain of mitochondria, but also in redox enzymes and for the purpose of transferring electrons. The problem with iron is that it can be very dangerous. Iron is a very potent oxidant, particularly when combined with hydrogen peroxide, which is not an unusual molecule. Hydrogen peroxide occurs in cells at concentrations of micromolar. So it is actually an abundant molecule, hydrogen peroxide. And together with iron, they catalyze production of free, or what people call reactive oxygen species. Some of them very dangerous, others not as dangerous as they. So the body requires iron, but it cannot have it wreak havoc. So it has to be very tightly controlled, not just by the amount, but also in how is it stored and how is it distributed in the body. So essentially, we don't want to have any free iron. We want iron always to be bound those two orbitals that can have unpaired electrons. They need to be coordinated in a way that they're not free, generate free radicals. And so we have lots of proteins that bind iron and try to keep it in safe formats in the body. We also absolutely require it. Without it, we very quickly run out of energy because we cannot transport oxygen. Our cells starve of oxygen and they cannot produce energy. On the other hand, if we have too much, we end up with all sorts of diseases that have been shown to have either depend on iron, such as anemia and some cardiovascular diseases, or others that at least iron is heavily associated with them. So we have not shown that is necessarily a causative effect, for example, in Parkinson's disease. But the truth is that Parkinson's disease, iron has a very important role. Free iron has a very important role in the disease itself. Even if it is not a cause of the disease, it is involved in the etiology. Another one, Alzheimer's. Alzheimer's has these plaques formed by amyloid or tau protein. These plaques are heavily binding iron. So if you hydrolyze those plaques, you get loads of iron come out of those fibrils. So again, this may be a side effect of those fibril formations. It may be that iron is catalyzing them. We know that iron has a role there, and that means many other diseases. This paper that I showed there from a colleague of mine does put together the evidence that iron is really involved in many, many, many things. And people may ask, so why is that? And by the way, one of them that it's involved, it's not a disease, but it's something we see naturally. It's called aging. Okay? We get old, partly also because of iron. And the reason for this is this reaction that iron enters, which forms free radicals. That's called a Fenton reaction. As I told you, ferrous iron reacts with hydrogen peroxide. And one of the things it forms, this molecule here, it's actually not a molecule, it's a radical, has one unpaired electron, it's called hydroxyl. It's the most potent oxidant that we know, apart from free electrons. Now, free electrons are not very common, not in solution. I know in wires they are, but not in solution. So it is really the most potent oxidant that is known. Now, unfortunately, iron can go back to the ferrous form, so it oxidizes, right? Goes to the reduced form to oxidize and create hydroxyl, but then that reduced form can go back to iron 2+, because of if superoxide is present, and superoxide is present when you have superoxide dismutase that regenerates hydrogen peroxide, and so you can, if you look at these three reactions together, only this one is enzyme catalyzed, these two are spontaneous, you end up with iron being a catalyst for the formation of free radicals. All you need is iron and superoxide dismutase, and superoxide, of course, and you have continuous production of hydroxyl, which is a molecule that is so oxidizing that it doesn't have a preferential co-substrate that it oxidizes. It oxidizes the first thing that it encounters. So it is a reaction that, so if it's close to a protein, it will oxidize a protein. If it's close to DNA, it will oxidize DNA. It will create mutations. If it's next to a lipid, it will form lipid hydroperoxides, lipid radicals. And so this is the reason why iron is so dangerous. Iron alone is not, but it's iron together with oxygen. Okay, so I don't want to spend much time with why it is dangerous, just need to alert you that it is, and that's why the body has all of this stuff I'm going to talk about, which is how to keep it safe, and also how to use it for what is needed. So we humans have about, I think around 4 grams of iron in our body, of which about 60 to 70%, depending on the people, are actually located in our red blood cells. Red blood cells also happen to be the cells that are most abundant in our body. They outnumber all other cells by orders of magnitude. So we are essentially made of red blood cells. We're made of water, but if you look at cells, that's the largest number of cells we have. And those cells are designed to just transport oxygen from the lungs to all the parts of the body. And iron in the form of hemoglobin, is what does that transport. And so there's about 2.5 to 3 grams of iron in the red blood cells. The red blood cells are magnetic. If you do microfluidics and you put a magnet, they are actually magnetic. That's how much iron they have. Now, the red blood cells don't live forever, actually because they have iron and oxygen, so they have a lot of oxidation. So in our body, they live about 120 days on average. There's actually quite a large variability on that. I will later talk about variability. But they kind of die quickly, so it's about 120 days. So they have to be made all the time, and they're made in the bone marrow, inside our bones. And this process is quite dynamic. Also, the cells that are old, they need to be recycled. We don't have a way of excreting iron. Iron is only loss from epithelial cells, so it's a very slow loss. So the body recovers the iron from the old red blood cells, and that's done in macrophages, mostly in the spleen, but sometimes also in the liver. And those macrophages, basically, that's why they call macrophages, they really eat red blood cells, they engulf them, and they completely hydrolyze them, and they pull out the iron, and they keep the iron and release it when it's needed. When they release the iron, it goes back to plasma. Now, this cycle has a rate of about 20 to 25 milligrams per day. There's iron is circulating around this cycle about 25 milligrams per day. However, we eat only about 1 to 2 milligrams per day, and we lose also about 1 to 2 milligrams per day. So if you just to see, the amount of iron that is circulating in this erythroid system is much larger flux than the flux of iron coming through and getting out of our body. On normal people, on some diseases, this is not the case. So there is also some iron in the liver. The liver seems to be a place where iron is stored. We thought of storage. Actually, from my models, I can formulate the hypothesis that the liver is actually detoxifying iron. It's not storing it. It's just getting it away from other more dangerous places. And so there's about some cycle here as well. And this is the plasma. Now, this is the picture of iron regulation that we knew up until 2000. In 2000, a very important piece was discovered called hepcidin. It's a small peptide hormone. It was actually first thought of as an antimicrobial peptide, but it's not. It's a hormone that regulates iron. And what hepcidin does is that it binds to a protein that is the only protein that can export iron from any cell to the blood, called ferroportin. It's a solid carrier protein, and it transports iron out of cells, not in, only out, and it's the only one, and hepcidin binds to it, and when it binds to ferroportin, they both get degraded, and so more hepcidin means there's no exit of iron from cells. And why is this important? Because to absorb iron in the intestine, we use ferroportin. So iron gets into the cells of the intestinal wall, and then from there, they go into the plasma using this protein ferroportin. So if we have high hepcidin, we no longer absorb iron. But ferroportin also exists in the liver, so if there is higher hepcidin, then the liver also doesn't export iron. And there's also ferroportin in the macrophages, so if there is high hepcidin, the macrophages also don't export. So what happens is, when hepcidin is expressed, the iron gets immobilized. It gets stored in the cells where it was, but it doesn't leave. Okay? And particularly, it also doesn't get absorbed. So people that have high hepcidin levels will not absorb iron. And in fact, this is one disease called anemia of inflammation, because inflammation also causes elevated hepcidin. And then those people will actually they have enough iron, but the iron doesn't get released, and so it doesn't get to the bone marrow, and therefore at some stage they don't have enough iron in their red blood cells, even though they have lots of iron in the macrophages, in the spleen, lots of iron in the liver. Okay. Now, hepcidin is itself synthesized in the liver, and it's the liver that senses hepcidin and that then releases it. So the liver seems to be important in the control of iron. So we started to want to model iron in the whole human, and we thought, okay, let's use the bottom-up approach. We start with modeling cells, the important cell types. Then we go from there, we build, using the cell types, we build models of organs. And then from that, we can build a model of a person. Okay, this is called the bottom-up approach. And we started that way. And the most, well, we first actually started by, in this paper, started by putting together just a molecular network that was known of all of the things that are involved in iron, all the proteins that bind it, all the proteins that transport it, the ones that store it, the ones that let it in and out, the hormones that are related, etc. Everything, all the transcription factors, the regulatory proteins of the kind. So we made a little nice diagram of iron network, we called it. And then we also projected that onto different cells by figuring out whether the proteins are expressed or not in those cell types. And so we had also maps of iron network for specific proteins. And very quickly we figured out, well, the liver is so important, let's start with a model of a hepatocyte, a liver cell, which is what a PhD student of mine in Manchester when I was still there did. And so that's a very nice model of the induction of hepcidin. It includes the receptor, which is actually a transferrin receptor that senses the amount of iron that is bound to the transferrin protein in the plasma. And more iron, it induces hepcidin, because of course what the body wants to do is not to absorb more iron when we have enough. So this is a classical negative feedback loop. If we have enough iron, then the entry of iron gets inhibited, and so that stabilizes the iron level. And so we did that model, and we published that a few years ago, and so that's one level. The problem with this approach, we then found out, is that that model gave us the response of the liver to iron and producing hepcidin. Now, what happens when there's production of hepcidin? Well, the liver itself no longer releases iron, and that we could represent because our model is of a liver cell. But also, there's no iron absorbed. So the plasma iron will tend to start going down. Now, our model cannot do that. So we had to artificially start manipulating parameters to simulate what happens in the body. And we very quickly figured out that this bottom-up approach is not ideal. It's not the best way to go. Well, that was my conclusion. So let's think again about this problem. We need to have the whole body response before we know what each specific cell does, because the whole body needs to respond to it. So we went the other way, the top-down approach. And the top-down approach starts by creating a model of the whole individual, and it's a model at low resolution. So we only represent organs and how much iron is in the organs, not which proteins they're bound to or whether they're in ferritin or free iron or whatever. But we care about all of the organs that matter. And then we go down from there. Then we use models of cells, we substitute them for the particular organs. And so that way, essentially we have a scaffold where we can then plug in the various lower level, more high resolution models of the various cell types. That's what we're doing now. So we started with a model for mice. And why mice? Because you can do things in mice you cannot do to humans. And part of this work has been already published, so the first part you'll hear is published, but the second part is not published yet. And this is an OD-based model. It only really tracks only the proteins that are absolutely necessary, which in this case is transferrin, the protein that transports iron in the blood, ferroportin, which is the protein I mentioned already that allows iron to come out, hepcidin, the hormone that inhibits it, and this thing called NTBI, which is the free iron in the blood. It's called non-transferrin bound iron. It's a big name in the iron field, but just think of it as free iron in the plasma. And we used data that had already been published of an experiment that was done where lots of mice were sacrificed. I mean, poor things. And these mice the experiment consisted of the following. They had 3 sets of mice. In one set, they give them the adequate amount of iron. This is representing the iron in their diet. The other ones have low iron diet, so they are iron deficient. And then the other group, they gave them too much iron, so they have iron overload. And they fed them for a number of days, 35 days. So 35 days, they're very happily living, eating their diets with too much, too little, or enough iron. And then after 35 days, they inject an iron, radioactive form of iron, isotope 15, iron 59, which they can track. Okay? The idea of using radioactive iron is that now you can track, you can measure the fluxes of iron through all these organs that I mentioned. And they did that, and then for 28 days, they followed the radioactivity, and they followed it in the different organs. And how do they do this? Well, every few days, they kill a bunch of mice, they dissect them, they get all their organs, and they carefully measure the radioactivity in each one of them. This is why you cannot do this with humans. You can actually give radioactive iron to humans. You can do it in such a small amount that is less radiation than an X-ray. What you cannot do is kill them and then start measuring all their body parts. Or mice actually have that done. So what we end up is with a picture of iron in the different organs, and that's what our model represents. And so this is kind of a high-level view of the model. Has plasma in the center, that's the blood. Let's think of it, the liquid part of the blood, the solution part of the blood. It has also, of course, red blood cells, the cells that contain the most of iron. Has bone marrow, because that's how iron goes. Iron goes from the plasma to the bone marrow. Then bone marrow produces red blood cells. Then the red blood cells get eaten in the spleen, and the spleen eventually releases iron again. And the liver also takes in some iron, and it produces this hepcidin. And then we have to represent the rest of the body, because we don't care about at this stage, we don't care about the brain and the spleen and the heart, but they have iron, and we have to account for it. So we just lumped everything together and call it rest of the body. And the small intestine, the duodenum, is needed because that's where iron gets absorbed. So that's the model. So we just had the model contains a number of equations, which doesn't really matter. We set it all up in Copasi. We do parameter estimation. And we add a few constraints just because there's too many parameters and not enough data. We added a few constraints. We know the iron in the mice is around 2 milligrams, so we constrained it to be within 10% of that, plus or minus. And we also know how much transferrin saturation, some other iron parameters, so we use those to screen models. We trained many models, and then we pulled out the ones that fulfill this criteria. So that's basically, for the machine learning people, that's our defense against overfitting, essentially. It's a little bit of regularization done in a different way. And so here it is. We fitted the normal data. So what you see there is the simulation are the continuous lines, and the data are the dots with error bars where they're available. And by and large, it's a pretty decent model. It represents what happened to these mice. These are the ones in the normal diet. Okay, these have the adequate amount of iron. So now, what happens when we try this on the iron deficient? Now, we don't change the parameter values. The only thing we change is the amount of iron going in through the diet. So there's only one parameter changed, the diet. Everything else has been calibrated to the normal. So this is somewhat a validation already. Does it work? Well, it kind of works. It's not terrible. It's not as good as the other one, but that's what modeling is. And given the error bars, as you can see, there's some pretty large error bars here, because the mice are all kind of different. Less different than we are as people. As individuals, mice are actually much more genetically uniform. That's why we use them. But they still have a lot of variation. So it's not so bad. So it works for low iron diet. Also works for anemias. But does it work for the iron overload? And the answer was no, it doesn't. And to cut a long story short, because details are not so important in this topic of this conference anyway. So the point is, our model overestimates how much iron goes into the red blood cells when there's iron overload, and it underestimates how much goes into the liver. So the model is really not working, because when we eat too much iron, what happens is the excess iron goes all into the liver. We don't make more red blood cells because we have more iron. Actually, it's bad to have more red blood cells. More red blood cells gives you stroke. Your blood becomes very viscous and you have problems with it. Now, it's an advantage if you live very high up in the mountains, so people in Peru, probably the people who are adapted to the high altitudes, have more red blood cells than we do. And that's why when they go run marathons, they normally win. That's why the Ethiopian people who live very high altitudes run very nicely, long distances, because they have lots of red blood cells. They have, unfortunately, a lot more strokes than we do. So there's not, nothing comes for free. And that's the problem. So the body doesn't want to make more red blood cells just because there's more iron. It just needs to tuck the iron away. And our model didn't do that. Our model was like, oh, more iron, more oxygen to be transferred. So that's wrong. So let's go for the next step. But before I do that, this is the important thing. I always give this slide to my students. Our model is wrong. But then again, all models are wrong anyway. This comes from George Box, a famous statistician, that quoted, all models are wrong, but some are useful. Then later on he even said, it doesn't matter if a model is wrong or right, what matters is if it is useful. And so what I'm trying to point out, and I had to make this case very strongly to the reviewers of the paper, who didn't want to approve it, because it basically said, oh, you're just showing your model doesn't work. The model is useful because it pointed us to how do we need to fix it. What is wrong in our thought? What's missing? And what's missing is there's iron going too much to the red blood cells and not enough to the liver. So the regulation is missing. And by the way, this also exposes a hypothesis that people had that hepcidin was the master regulator. Hepcidin it's all that coordinates all of the iron stuff, but clearly doesn't, because hepcidin alone doesn't allow us to reproduce this. So we had to add two more things. And we added erythropoietin, which you have heard as EPO, which is another hormone that stimulates making red blood cells. And more importantly, when we have too many, it goes down and the red blood cells don't get made. Okay? When we have too much oxygen, erythropoietin is inhibited, and therefore we don't make more red blood cells. That's how we not have a stroke. Of course, when we are low in oxygen, erythropoietin goes up, and the bone marrow pulls all the iron that it can, competes with the rest of the body, and says, you guys may need iron, but I need it more than you, and so I get all the iron. So we added that, and we added another protein that has been discovered to transport free iron into the liver. Okay, it's a transporter called ZIP14. It's actually originally thought to be a zinc transporter. It turns out that it's probably a manganese transporter, but when there is too much free iron, it allows iron to go in. So we added these two things. So this ZIP14 is this blue arrow here that allows NTBI to go into the liver, and now we added erythropoietin and all of the regulation. And more importantly, erythropoietin is activated by, it's inhibited by red blood cells. More iron in red blood cells, less erythropoietin. But erythropoietin stimulates the production of red blood cells. So there's another feedback loop here, another negative feedback loop here. But also erythropoietin inhibits hepcidin. So these feedback loops are no longer independent. They're kind of intertwined. So this now, as a dynamical system, becomes not completely trivial to understand how it works. So modeling is good. Anyway, our new model, does it work? And the answer is yes, it does work. This is the new model. This is the old model here, this yellow line, and then the black line is the new model. So you can see what our model does now is because there's more iron, but there's no lack of oxygen, erythropoietin comes down, and so there's not more production of red blood cells. What there is more free iron, but because the liver can take the free iron in, then the iron ends up in the liver, which is exactly what happens in the physiology. So we were happy with this. We said, okay, how can we validate it now? Because now we've used all the data, right? Now we've used the three data sets, the normal, the deficient, and the rich, to calibrate the model. So no longer becomes validation, right? So we try to be good in this, so we have to have independent validation. And we did validate it against data that exists for all sorts of diseases that we did not program into our model. But our model reflects exactly what happens in those diseases. And so for us, that's validation. Just as an example, beta thalassemia is a disease where people don't make one of the types of hemoglobin, and so they have problems. Their red blood cells don't live long, and also that's one of the problems. They have anemia, and the second problem is they accumulate a lot of iron in the liver. It's an anemia that contains iron overload in the liver. And in our model, we simulate beta thalassemia by changing only two parameters. This 3D plot here shows two parameters. By the way, this was done with parameter scan in Copasi. So two parameters. One of them is the efficiency of producing red blood cells, that is the rate at which iron goes from bone marrow to red blood cells. And the other one is the lifetime of the red blood cells. And by changing those two parameters, we can go from normal, which is this yellow region, to the kind of orangey region, which is so-called beta thalassemia minor. It's a mild form of the disease. It's not really that mild, but it's not the severe form of the disease. And then thalassemia major, which is the very severe form. And it turns out that it's not trivial because they're not really continuous. In this variable, they are continuous. It just goes up and saturates. But on this variable, these are relative changes of one of the variables. In thalassemia minor, you have overexpression of that variable, and thalassemia major, you have underexpression of that variable. But we can show that just by changing those two parameters, we can simulate the disease. We can even ask which of the two parameters is the more important one. And it turns out that it's the inefficient erythropoiesis. Just changing the lifetime of the red blood cells, making them live shorter, would only cause thalassemia minor. You would never have thalassemia major. I actually have to have inefficient erythropoiesis to get thalassemia major. So this is a way, again, that shows how a model can enlighten us about the scene. This is a useful model because it's obviously wrong. If I go into details, there are things that probably are wrong if I continue probing it. But it allows us to immediately start thinking about the system. And then we can do all kinds of things with this. So we can say, for example, how can we simulate some interventions in some diseases? So hemochromatosis is a genetic disease where people accumulate lots of iron. And the only efficient treatment we have is to give blood. Those people, we have to take blood from them. Because blood has most of the iron. So if we take blood, we take iron out. And there are different schedules to take their iron out. We can do it weekly or 2 weeks, or we can take out 10% or 15% or 20% of the blood. And we simulate and we say that 10% doesn't bring you back to the normal level, but both 20 and 15% bring you back to the normal level. So maybe you should only take 15% out. So this shows that we could use these models to optimize treatments. I did this by hand, but I could have used optimization. And this here is just to show if you ever think of using EPO to get more efficient, don't do it. Because apart from all sorts of other bad things that EPO does, it will overload your liver with iron. So most likely you will have cirrhosis at some point. Maybe you have other bigger problems, but one of the side effects of regular injections of erythropoietin is that you get accumulation in the liver. And as you can see, the peaks go higher and higher and higher, even though we're giving the same dose of EPO. It's a cumulative effect. It's not, once the iron is in the liver, it doesn't really come out. Okay, so now to close this, let me move on to humans, because we really don't want to treat mice. NIH would never give me money to come up with a disease to treat mice. Particularly these poor mice are all mutants anyway, the ones we use in labs. They're not even like the normal mice. So we want to use this in humans. And now, I was told by reviewers, and in fact I believe the same, that we have to do models in mice because there's not enough data in humans. Well, there isn't if you don't look for it. When we started looking for it, there's surprisingly quite a lot of data for humans. And it turns out that it doesn't even map with the data that exists for mice. We have some type of very high quality data with mice models. We have lots of data on other things that no one measures in mice that we measure in humans. So we have different sources of data. It became a very interesting thing, as you will see. So the roadmap essentially is to go from a mouse to a human, we first of all have to resize the animal, okay? I mean, mice are mammals like us, they are rodents, but they have many things in common. Let's assume, as we will later, we'll ask that question. For a moment, let's assume that people are just big mice. Our livers are bigger, our brains are bigger, our hearts are bigger, etc. So let's rescale the organs, the important organs. Fortunately, we don't have the brain as one, because that doesn't scale in the same way the other organs do. Let's rescale the organs to the size that they exist in humans. And then let's recalibrate the model. So we will need to have some data from humans, but of course we don't need it for all of the organs, because we already have an equation structured. So we need some data from what can be measured. For example, the parameters in the blood, it's easy. People can give blood. That doesn't kill anyone to give a little bit of blood. Not like giving a little bit of your heart, that's more problematic, right? But giving a little bit of blood, it's okay. So we can measure some things and recalibrate the model now that we have also resized the volume sizes. And then later on, we will have to deal with issues of variability. That is, I am different from you, and I'm different from you, and you are different from him, and he's different from him, and we are all different. Okay, so if you're going to apply these models to specific individuals, patients in need of some treatment, that we want to use the model to optimize the treatment, we're going to need to be able to calibrate the model to each one of you. And also, we're going to need to be able to represent the widespread variability that exists. So we are starting to deal with that, and later on we want to think about how to personalize the model, what to fit, what parameters do we fit. That's not so simple. Now, organ size is not a problem. Some people have a boring life. Coroners, the doctors in hospitals that have to officialize and do the autopsies and figure out why people died, they have a very boring life, and some of them, they don't do a lot of research, but some of them say, okay, what can we do with this? Let's look at the people that died of accidents. That is, they're supposedly normal. They didn't die because they had a disease. They died because they had a car accident, or they got stabbed by someone else, or they got shot, or whatever. And let's measure all the organs of those people. And there's enough people that die this way, of many ages, of many sex. So there's several papers that exist where thousands of human bodies were carefully measured for their volume. So we have distributions of volumes of livers and hearts and brains and spleens and everything for humans, which we don't have for mice, by the way. So there's the question, is a human a large mouse? And the answer is mostly yes. So here we have the sizes of human organs, the ones that we carry in our model, and the proportion that they have of the body. And what we did was we looked for how much different is the mass of a human from a mouse, and we scaled all the organs by that proportion from the mouse, and then compared with what people have measured with the humans. So this is what we scaled up, this is what is measured. And they're not so different, apart from the liver. It seems the liver is really different. We have smaller livers proportionally than mice have. Mice have much bigger livers proportionally to their mass. Apart from that, everything else is more or less in the range. If you look at it, we would have not made a big mistake if we just went with this data. This was made up. And these are the proportions of the total mass of each of them. So anyway, we could resize the organs. That was great. So now we need some data to recalibrate the model. We found some experiments where people have been injected with radioactive iron, just like the mice did. And then they measured some parts which were easy to measure, like the blood, the bone marrow, because you can measure the radioactivity very close to the bone with a Geiger counter. And the liver as well, because there's enough iron in the liver to get a signal. You don't have to cut the person open. You can actually measure it. So there are some data for this. And interestingly, the data cross several decades, starting from the 1950s, 60s, 70s, and even 2006. This is actually for the hormone hepcidin. And using this data, they map very nicely. So here, they're data from the 4 experiments. These first four experiments are all mapped there with different colors. And you could see they line up very nicely, which is nice to know, that people can have measured this stuff in the 1950s and then in the 1970s, and the data is still pretty much accurate. Okay, so it looks like our bodies do work more or less reproducibly. We kind of knew that, but. So here it is. We recalibrated the model to the data we had. In this case, we also have data on hepcidin, which no one ever measures in mice, okay, because it's expensive. You have to use antibodies, and in fact, we only have antibodies for the human hepcidin. We don't have antibodies for the mouse hepcidin, surprisingly enough. So we had some data in humans we didn't have in the mouse, which the reviewers still don't believe me. So we have a preliminary human model based on the mouse model. We could have not done the human model without the mouse, but actually now we could go back and improve the mouse model from the human because we have this data that only exists in humans. So I think there's a very important lesson here, is that you should be able to use so-called animal models and then move around with human models, but this is not just a one-way street. There is a lot to be gained by iterating the two. Now, just to finish the issue of variability. So eventually we want to have a model that can be recalibrated to every different person. That model must be able, first of all, to represent the range of behavior that we see in different people. So what is that range of behavior? So here we are starting to look for papers that show us variation, measurements on single individuals and variations about them. And it doesn't matter the details of them. There are several variables of our model that are included in these various papers, some of them old, some of them very new. There's a number of sources for this. And so we started doing that. I have a very talented undergraduate working in my group just for 2 months. She's almost finishing. She's done a really great job. She's actually a nursing student, a nursing undergraduate. She knew nothing about computers. She's really great. So that was good validation for me. So she collected lots of data. These are in hepcidin in the x-axis and the serum iron in the y-axis. And if you ignore the green data points, which are from a particular disease, people that have brain hemorrhage, if you take that away, you see the data is very much a pretty nice slope. More iron in the serum, more hepcidin, and that's kind of a part of that feedback loop. It's hard to know which one is causing the other, but it's a feedback loop. And now in the model, we started changing parameters, figuring out what parameters could we change, and we kind of get at least the same behavior. If you ignore the green, I should have not plotted the green, but if you just look at the red and the blue data points, they're nicely sloped, and these are sloped. We haven't got the ranges completely correct yet. But what are we changing? We're changing two sources. One is environmental, that's the amount of iron absorbed. Different people eat different diets, they have different amounts of iron that they absorb. That's just one difference right away from everybody. Your breakfast was not the same as mine, etc. Second one is genetic. That is, how much does the body produce, how much hepcidin does it produce for the same amount of iron in the blood? Because that could be different. That's the expression that has a signaling pathway that controls hepcidin, and the parameters of that signaling pathway may be different from people to people. We may have SNPs in our different genes that eventually make those parameters different. So we just change those two, and with those two alone, we can already kind of get the range of behavior. As I said, the particular ranges need to be calibrated, but she's only worked for a month and a half, so I think this is going in the right direction. So this gives us some hope that we will be able, with the model, to represent the variability. The problem will be the models of individuals, but we still, that's what is nice about this type of work, is that you don't have the answers, you have to find them. So I'm interested in finding these answers, and we don't know. But we have, again, a roadmap, at least a conceptual one, that first, before we create different individuals, we're going to try to look at different population groups. So, for example, men versus women, or women in reproductive age versus women post-menopause, because there's a big difference. Turns out one way to lose a lot of iron is losing blood, which is a common occurrence in women in reproductive age. So women are more anemic until they come to menopause. By the way, that's also the reason why women have less heart disease incidence. Postmenopausal women have exactly the same incidence of heart disease as men. The reason that women don't have more is that they have less iron. They're slightly anemic during their reproductive age. But so we would go to groups like that, maybe look at some genotypic groups. Very hard to do genotypic groups with humans because we are very, very, very diverse. We have a lot of diversity on our genome. We're not like mice, where you can easily get BL mice versus other kinds of mice, and they're very different already, but very unique. We are all over the place. And then try to look at what parameters we can measure from people. So now the easy ones, the ones you can get when you go to your doctor and you don't care about, are things like your height, your BMI, which we can calibrate the organ sizes that way. Things like your hematological parameters, so come from blood analysis, those things people do. But then people will also be sequenced. So we have to think about what can we do with their genome sequence, because people are going to be sequenced. In the USA, there's thousands of people being sequenced already. And it's going to be in the hundreds of thousands quickly and probably millions very soon. Sequencing is very cheap. So we don't know what we can do with it, but probably is looking at SNPs and classifying SNPs and connecting them to parameter values. That's the hard thing to do, I think. So I think that's basically everything. I'm going to finish here and acknowledge the people who actually did most of the work. I've already acknowledged the team that built Skopazi. I need to acknowledge Jignesh Parmar, a very talented postdoc in my group. He built the entire mouse models. Well, there was a little bit of help from 2 undergraduates in the very beginning. They collected the sources of data to calibrate, etc. But then Jignesh really developed the modeling, and he's really a super modeler. I can only praise him. Now, 2 other undergraduates built the human model, and now Tram is the current one that's looking for the variability data. And of course, we had funding in my group from these 3 sources: the NIH in the US, the National Science Foundation that pay the undergraduates terms, and the British BBSRC that funds biochemical and biotechnology research. So thank you very much, and if there's time, I'll be happy to answer questions.
So we have time. Oh, sorry. We have a good time for questions. Sorry, might move this way. Yeah. Any questions? Maybe I will open the session of questions. I'd like to ask you, Pedro, is it possible to use a systems biology approach like this to infer new interactions? In this case, you have like the first error, so errors in the first model, and then you have some reported proteins or reported interactions that you could include in your model. But if it wouldn't be the case, and you say, okay, I must have something here, some novel interaction that it would prompt discovery of novel interactions. So how would this work?
Well, I think so. I mean, absolutely. I think that's really the utility of a model. That's what Box was talking about, that models have to be useful. And even if they're wrong and they were useful to lead you in the right way, that's exactly what we want, and they're tools. They're not the ultimate end. So in our case, those 2 things we've added, one of them is not controversial and it was well established. It's one of those things we say, why didn't we think about it before, right? The erythropoietin. In fact, for a long time, that's the only hormone that was known related with iron for many decades. But the ZIP14 transporter, it's actually not completely established. So so the people who have proposed it to be a transporter of iron into the liver, I think they will be happy with our model because they're still kind of establishing that that's the case. It is not a completely accepted fact that there is transport of free iron into the liver. Now that explains a lot of things if it's true, and in their papers they did this. And so this was actually a recent discovery. So we thought of adding entry of free iron because we saw very large accumulation of free iron in the blood. And that is not normal. People will get disease if they have that. So we thought, okay, maybe free iron goes in. And then my postdoc, Jignesh, he found that these papers that list ZIP14. Because we want to convince people that it's plausible what we do in the model, right? So once we did that, so actually we, quotes, discovered it by the defect of one model and to fix it. Now it turns out we didn't discover it because there are papers where it has been shown. You know, they have mutants. The mutants don't have any liver accumulation. They have a lot of iron in the plasma. Actually, their mutants look like our bad model. And so it worked out wonderfully. And I'm hoping, because that's under review right now, or the editors. I'm hoping that we will have not as bad a time as we had with the previous paper, because they're probably going to say that it's obvious, but I'm very happy. I think that's a very, very, very good way to use models. That's the best. That's how they can contribute to discovery. They're a tool just like microscopy or spectrometry or whatever.
Any other questions from the audience?
This one in the back there.
Thanks, Pedro, for your presentation. It was very interesting for me. And I was wondering if you are thinking on trying to conciliate the approach that you have top-bottom with this one that you presented, top-down, well, the
Bottom
opposite
up and
direction.
top down, yes. Well, I mean, I think we're doing that in a sense because, so what I did not describe is we've already tried, since we have the hepatocyte model, the first one I talked about, we already tried putting it in place of the liver. So we assumed, again, another assumption, that the liver is one big hepatocyte. It's very wrong, it's not correct. But just for the sake of argument, we have the liver being one hepatocyte that is scaled up to the size of the liver so that it gets all of the iron that's needed. And that actually works okay. The model has some, if we go outside of the normal range of parameters, if we push some parameters, the model has some problems with, we have some very fast oscillations and things that we think are spurious. But by and large, we're very encouraged that that will work. So we think that that's going to work okay. So essentially, it is top-down because we start with the top, but then it's bottom-up after that because we don't use the organ. We use first the cell types. The organs are more complicated. They require many cell types, so we will probably do that at the very end. So yes, I think I think these concepts of top-down and bottom-up are really very theoretical. They're very interesting from a conceptual, formal point of view, but in reality, nothing ever works exactly like that. You end up always with a mix. We had to use some proteins. I mean, even though we were talking about organs, we had to have ferroportin. Without it, this doesn't work.
Any other questions?
Thanks, Pedro, for the talk. That was very interesting. So my name is Pascal, and I'm from the University of Toronto. Mm-hmm. We work in the area of intraplaque hemorrhage in the carotid arteries. Mm-hmm. And one of the areas that we're particularly interested in is the haptoglobin binding with the free hemoglobin. Mm-hmm. And I was just wondering for macrophage reuptake, for those that are not familiar. And so I was just wondering, have you looked into that aspect of it for modeling, because that's would be very interesting for us.
Yes, no, not that particular one, but there, just like that, there's other things that are also very interesting. For example, the erythropoietin story. Actually, there's a whole range of different issues with erythropoietin, and it turns out those people doing erythropoietin, they figured out they also need iron, right? So it's almost like these things have to bolt together, and I think that's exactly the case. Like some of the data I showed with this brain hemorrhage. It shows a completely different slope between hepcidin and iron. That suggests that something is happening there that changes this feedback loop, right? At the moment, I don't know what to do with it, but it's quite interesting. So it points to the fact that eventually these things have to be linked. That's the real systems biology, right? Mm-hmm. Because we call these things different areas, right? Or different subject areas or different disciplines. But in reality, nature is all one, right? There's no difference. The hemoglobin doesn't know if it's part of the iron model or part of the other things that happen to it. So,
one more question, if I may. Just in terms of modeling, so one of the ideas that we've been working with, and we tested this in bunnies, it's to look at chelating agents for iron to see if whether or not that would improve IPH. Yes. And in the case of bunnies, it did.
It does.
But I'm just curious in terms of modeling, because of course the concern is doing chelating in humans, right? So
it's a controversial thing. I mean, we've looked at it. In fact, some of our validations include adding chelators. Chelators are controversial because the problem with chelators is that they have to work on very large. I mean, this is. Iron is not like small molecules, right? Because we have grams of it. We have large amounts of iron. So chelators have to work in a range where they have to the binding affinities have to be very different than molecules that are used for binding for other things. So the clinicians seem to have a problem with chelators. Now, better chelators are appearing because, by the way, because they have to be in millimolar concentrations, and most molecules in millimolar are toxic. Mm-hmm. So people are very careful about adding chelators. They only do it when they need to. But I think there are new more modern chelators appearing that have some of these side effects have gone away. Mm-hmm. And I think they actually look very promising for people like hemochromatosis, even though it's not used for that. Mm-hmm. I would say probably. The problem with so what chelators do, they change our model because they make iron be excreted through the urine, which is not a normal route of actually. no excretion. We lose iron because we lose skin cells and we lose hair and we lose epithelial cells. That's really the loss of iron. But with chelators, they go out in the urine. Yeah, I think there's a lot of research in that area. I think the chemists are really trying to get new organic molecules that can be in millimolar concentration that have no side effects and chelate iron.
Thank you very much.
Thank you.
Any further questions? So I do have another one. Have you performed sensitivity analysis to see what would be the most robust targets on this model, or do you really need to have to reconcile first the top-down and the bottom-up?
No, I mean, we've done a little bit of sensitivity analysis. It doesn't, I mean, this is a complex model, even though it looks simple, because it's only iron in different parts of the body. It is actually, dynamically, it's quite complicated. So it depends what you look as the variable of interest. So we looked, for example, at hepcidin as, you know, what makes hepcidin sensitive, and kind of got the answers of the things that look good. So we did that in terms of saying, is the model, does it look, sane? Is it kind of, does it make sense to what people already know? And it kind of, so we use sensitivity analysis for that. The problem with sensitivity analysis is that if we only do the local, I mean, it's very much dependent on the particular condition. So we would have to do global sensitivity analysis. What we have done is profile likelihood of the parameter estimation. Unfortunately, not all the parameters are fully identifiable, but that's always the case. Yeah. Interestingly, when we added the two, when we improved the model, it also improved the identifiability, which is also reassuring, even though the model is more complex. It has now more parameters and more variables, but somehow it's more constrained in some sense. So,
what about the stability? Is there base stability? Or
not that we have found. We haven't looked for it either, so okay. I don't know. I don't. I mean, the loops are complex. I mean, I could identify quickly two negative feedback loops, but if you look at the whole diagram, there are likely probably positive feedback loops in there. They're probably complex, but I think they might be. I don't know. But if we don't have them, then we won't have price stability. Right. Since I have not identified one positive, I have not tried that yet, but maybe. Okay.
The last question about the variability. Is this possible to say that the model or the biochemical pathway could be that variable between individuals that you can have different network topologies, or is like functionality?
I mean, I don't know, different network topologies. Depends what you think the network is, right? So if something is zero, you take the arrow out of the network. Right. Or do you have it there with a zero weight? If you have it there with a zero weight, I don't think the network changes. Right. But obviously, some things may be very low expression because of a disease or because of different characteristics. I mean, what we call disease is also relative. A person can be more athletic than another one, and we don't call the one that is not very athletic a diseased person. It's just different. So many of these are phenotypes, different phenotypes that are not necessarily disease. I don't think the network changes, but I look at the network as the arrows are everything that is possible, and then we put weights, which is how much is happening. And one of the weights could be zero. So I still see the arrow there. It's just that it's not being expressed. But the interesting thing with the variability is that it looks like there's many ranges of parameter values and works very well. We tolerate quite a lot of insult and are still normal.
Thank you very much, Pedro. If there are no further questions, yeah.
Thank you, Pedro. My name is Andrés Segura from Universidad Estatal de Distancia here in Costa Rica. And Rodrigo and some other colleagues have been working with capacity for a time. And one of the issues we found is that as we wish to increase complexity in the models, computational complexity is also an issue, right? So I would like to hear about what's your experience as far as computational resources and how cloud capacity, for example, or distributed computing has been helpful in terms of reducing the computing time of these complex models.
Yeah, so obviously that's a common problem everywhere, not just in computational biology, in all computational sciences. And we've created Condor Copasi and Cloud Copasi to come up with that. Now, these are methods to use cloud computing in a way that that we actually don't change the code internally, so not everything is parallelizable. Only the trivially parallelizable tasks. So for example, parameter scan is a very good example. It's trivial to parallelize because each run is totally independent from the other. So we've done those tasks, but the hard things is the parameter estimation. That's the task that normally takes very long time. And most of those algorithms are not easy to parallelize. I mean, they are if you want to go up to eight processing units or 32, but they're not going to scale. Parallelizing those algorithms will not scale to thousands because you get this Amdahl's problem, right? You get incredible numbers of communication. Most of the processing units will be idle waiting for messages from the others to move on. And we've tried that with simulated annealing, and very quickly you hit the limit. We combined it with genetic algorithms. We got a little bit further, but still we were like maxing out at 64 units. So it becomes, you know, at that stage, it's a lot of effort programming to only get that. So we are really focused on the trivial things. Any tasks that are independent, we've messed with that. But unfortunately, these optimization algorithms, not so much. Some of them have parallel versions that we will try at some stage to incorporate, but so far we've got lots of things to do. We've stayed away from parallelization. I had a very for a while I was a professor in a computer science department, and a colleague of mine who was a person involved in parallelization, he kept telling us that you really only parallelize when you know that you're gonna get bang for the buck, because otherwise you can really bury programmers' time, I mean developers' time, to get something that only speeds up things by threefold when you have 20 processing units, and that's not worth it. You end up better parallelizing the tasks that are independent. We have a little bit of parallelization already because the linear algebra routines, they use OpenMP. So if you have a multi-core, when the system is doing eigenvalues and things like that, it's actually using all the cores. So it's a little faster on a quad core than on a single core machine, but you probably won't notice that. Those tasks don't have a very strong impact on the computing time.
Thank you very much.
Unfortunately, there is not a good answer for that one.
Very good question. Anyone else? And we have to thank Pedro for such a nice presentation and contribution. All right, so we are closing the opening ceremony. And I would like to thank all of you here for being here, for participating on this IWOBI. I would like you to enjoy the talks, and I just would like to make some final announcements here. First, the location of the session rooms. You just go right, and then again right, and then take the hall all the way. We're going to a coffee break right now. There you will see the dining room, and there we will have the coffee break. Right in front of that, there will be the session rooms. Please note that the numbers have been shifted regarding to the agenda. So they're shifted by 1. So the A15 is no longer A15, but it's A16 and so on. Also, the password for the Wi-Fi is noted down on the doors on the session rooms. So there you will see the pages printed with the information. Please also remind that we have a WhatsApp channel for communication. Any question you have, you can write it there also. And we have a digital agenda on our website. And let us not forget, we have the social event today in the afternoon. This will be at Los Lagos Hotel. And the dress code, if you want to go to the thermal waters and dip in the water, you have to take bathing suits, of course. And this will be, the buses will be departing at 4:30 p.m. for that event. So thank you very much. Have fun and enjoy IWOBI 2018.